Hi everyone. Today we're going to talk about something crucial in cell culture, record keeping. Proper documentation is key to maintaining high-quality results and ensuring reproducibility. Accurate records not only help you track your process, but also make troubleshooting and improving protocols much easier. So, what exactly should you be recording? There are several key areas that are crucial to keep track of in every cell culture process. First, you need to document your cell culture passages. This is one of the most important aspects of your records because it tells you the health and age of your cultures. You should note every time you pass your cells, including the date, passage number, and any observations about their growth, morphology, or overall condition. Tracking this helps you monitor the life cycle of your cultures and avoid problems with overcultured or aged cells. Second, keep track of your cleaning and disinfection procedures. Cleanliness is absolutely critical in cell culture, and you must maintain a detailed log of all cleaning activities. This includes wiping down biosafety cabinets, incubators, and work surfaces with disinfectants, and recording the types of disinfectants used along with the dates. By maintaining this record, you ensure that your workspace remains sterile, and you can identify any gaps in your cleaning routine if problems arise. Third, make sure you record information about the reagents and media you use. This includes recording lot numbers, suppliers, expiration dates, date opened, and any tests for sterility, mycoplasma, or endotoxins. Using fresh, certified media and reagents is critical to your culture's success, and keeping these details on hand allows you to trace any potential issues back to the source, especially if a contamination or problem occurs. Additionally, it's just as important to document the maintenance and calibration of your equipment. Regular checks on incubators, centrifuges, biosafety cabinets, and other instruments ensure they're functioning properly. Be sure to log any maintenance or calibration activities so you can ensure optimal conditions for your cultures and avoid equipment failures that could compromise your results. Also, always record environmental conditions, including temperature, CO2 levels, and humidity inside your incubators. It's essential to monitor these factors closely, as even small fluctuations can affect your cells. Note any changes in your culture environment, as well as observations like abnormal growth patterns or changes in morphology, because these could indicate problems with the culture conditions or potential contamination. In summary, keeping accurate, detailed records isn't just a good habit, it's an essential part of ensuring successful and reproducible experiments. By documenting everything from cell passages to environmental conditions, you'll have a reliable reference to troubleshoot problems, improve your protocols, and ultimately ensure the success of your research. Thanks for watching and good luck with your cell culture work.